chemistry. We have to design the molecule and we have to synthesize the molecule based on docking studies. And once the molecule is good, that we have to go for analysis, that is the purification part. And third part is the ecology screening part. And next part is polymorphism, that is nothing but Sutix, making tablets and tablets and capsules. Polymorphism, crystal structure, novel formulation, and other techniques we have to follow. And next one is pharmacy is the health profession that links in between the health science and chemical science. Okay?
Okay, sir. Proceed.
Drug. A drug means, what is the definition for a drug means? Okay, till now I have defined synthesis, design, synthesis, docking, screening, outlines, definition for drug discovery I told. Now, what is a drug? Drugs are two types. According to chemical sense, synthetic drugs are there. According to biotechnology, vaccines are there. That is called biotech-based drugs only. Synthetic drugs, you know very well. The molecule, you are synthesizing the molecule and using screening, using that methodology I told. That's all called synthetic drugs and natural products, one category. And second one is biotech-based, is like antibodies, monoclonal antibodies, vaccines, using RNA technologies. This is completely different. So I will define the drug definition. A drug which is a chemical substance which is required for the body. So in the body, basically two things are there. One is antibody, second one is antigen. These two are present in our body. Antibodies are immune centers. Protein-based antibodies are police centers in our body. In our entire body, police centers are there. Antigens are microbial cultures, or any foreign body, any unwanted microbial culture, or any virus, or bacteria, fungi, anything is there in the body that is called as a foreign body. That is called as an antigen. So in the body, two things are there, antibody and antigen. Antibodies are, whenever the antigens comes in the body and antibodies are firing them, like a police center, it is firing them. It is killing that antigens. That is the function of, antibodies in our body. So regularly, antibodies are present in our body and kills the antigens. That is a regular phenomena. If antibodies are not there in the body, then our body will be destroyed. For example, death body, you keep it outside. Within the 2 days, it will decompose, or within the 1 day, it will decompose because antibodies are not working properly. Suppose in the active body, live body, antibodies are working. So already God has given the antibodies in our body to kill the bacteria or virus, but sometimes antibody activity is not sufficient. Antibodies are not sufficient in the body to kill the microorganism. Then the chemical substance which we are taking into our body is called as a drug only. Antibodies already working in the body. Sometimes which is that force is not sufficient. The external substance, chemical substance which we are taking from outside into the body is called as a chemical substance, is called as a drug only. Basically, chemical synthetic drugs are like that. What is the phenomena of chemical synthesis means? That is how to synthesize the new drug means I will define. You take the bleaching powder, put in the water, CaO, Cl2, calcium will go, chloride will go, nascent toxin will come out. Nascent toxin kills the bacteria. That is the phenomena. Very simple phenomena. In the water, bacteria is available. Wherever you see in the nature, bacteria is available. So therefore, we are to kill in the water bacteria, we are using the bleaching powder. Nascent toxin will come out. Nascent toxin only will kill the bacteria. It is oxidant. That means all oxidants are power centers, like nitrogen, phosphorus, oxygen, sulfur, fluorine, chlorine, bromine, iodine. These all non-metallic groups are nothing but oxidants. Oxidants means power centers, which gives lone pair of electron. Lone pair of electrons means current only. You touch that current, it shocks to the body. Like that, you touch the oxygen, nascent toxin, it gives some power, which kills that bacteria. Same thing like nitrogen. All nitrogens are having lone pair of electrons. It is nothing but flow of electrons is current. Flow of electrons is current means that electrons goes into the bacteria, which kills the body. That is a basic mechanism of the drug activity. So therefore, nascent toxin, nitrogen, phosphorus, chlorine, fluorine, all are Non-metallic species are oxidants only, which are power centers only, which kills the bacteria or any foreign body, or nothing but antigens are killed by these oxidants. In the periodic table, among all the elements, fluorine is the toughest, okay, non-metallic species, which is oxidant. A beautiful oxidant is the fluorine. So fluorine we are inserting in the molecule will become the cancer molecule. For example, omeprazole, esomeprazole, lansomeprazole, pantoprazole, you take, nitrogen insertion is there. Nitrogen is pyridine ring. Pyrrole rings are there in the molecule structure that can be acting as, okay, ulcers. For example, oxidants you are putting, that is one kind of oxidants. Like nitrogens, many nitrogens you are putting in the molecule that can act as a drug only. We are enhancing the nitrogen molecule. And you are using the molecules, making the molecule and using for the body. This is the basic drug mechanism. How the drug acts on the body means only simply in the benzene ring, we are taking in the benzene ring in all the molecules. Benzene have lone pair of electrons, 3 pairs of lone pair of electrons are there, 6 pi electrons are moving in the benzene ring. This is the generating the current only in the benzene ring. 1 benzene ring, 2 benzene rings are, okay, 5 membered rings are having the lone pair of electrons generating the current. That current, along with that current, you are adding the nitrogens or fluorines or chlorines that will make more number of electrons, which generates a power. A power center kills the bacteria or virus. That is the basic mechanism of the synthetic drugs only. Now, non-metal is such a powerful tool in the drug discovery mechanism. Generally, earlier we have prepared many nitrogen containing heterocyclic compounds by myself. 1,000 molecules we prepared. Now we are inserting the fluorine molecule and fluorine atom, and we are enhancing the activity and screening for anti-cancer. That is the logic of anti-cancer structures. You see, all anti-cancer structures you see, that is the fluorine center is there, fluorine atom is there, otherwise chlorine atom is there. These two are powerful. You see the chlorine nature, by nature outside, it is the toughest smell, which goes into the bacteria, which is very tough chlorine molecule smell. Fluorine also, if you touch the fluorine, our skin will be affected. Our fluorine, chlorine, bromine, these bromine also very tough, you know that. And iodine, these are the toughest accidents. Those who are trying to insert in the molecule, non-molecule, enhancing the activity and using for the cancer activity, that is the drug discovery program. That is all synthetic drugs, what I am telling. Second one is biotechnology based drugs are, these are nothing but antibodies I told in the body. That means God created a body. God asked an engineer to construct a body. Then engineer told, I need 23 pairs of chromosomes to construct a human being body. Then that engineer asked the God, so give 23 pairs of chromosome. I can prepare a human being body. Then God has sanctioned. Then 23 pairs of chromosomes are taken by that engineer. He constructed the body and he made a body, and all our diseases are responsible. Our diseases are all from defect of 23 pairs of chromosomes only. That is the basic funda. Basic funda is any chromosome is defected in the body, then ultimately that reflects as a disease, permanent disease. For example, diabetic is there, cancer is there, such a permanent disease are there, like corona is there, such a disease are there. Some DNA defected mutation undergoes for mutation. That means all 23 pairs of chromosomes are responsible, means one chromosome is responsible for our color, one chromosome is our intelligence, one chromosome is our body buildup, one chromosome is our diabetic nature, one chromosome nature. Like in the body, 23 chromosomes are responsible for construction of body. That antibodies are safe in the body, then everything is safe. That is a biotechnology concept. You take liver is not working properly, you wash the liver. You take nutritional food for the liver. You take diabetic is there. For diabetic, that whatever the responsible, it is insulin releasing, that is pancreas is there. Pancreas not working means insulin defect will be there. Then pancreas is not working means wash properly pancreas or activate pancreas properly, or you take the nutritional food which is required for pancreas, then it works fine, it works very nicely. So all body requires nutritional food. You take the nutritional food, required exact food, the antibodies are developed in the body, antibodies are active and healthy means you will not get any disease. That is the biotechnology concept. Basically, the biotechnology concept is You take a milk and, okay, take a small curd and put in the milk. Then what happens? All milk can be converted to, okay, curd only. Like that also, antibodies you take from the body. That antibody you take and RDNA technology, use that and keep it in the media and convert the antibodies of diabetic responsible, okay, anti-diabetic responsible. Pancreas protein is there. I take the protein is not working. You take that protein, a protein recombinant technology, you make the many proteins of antibody. That antibody will work very nicely in future. Whenever you are shortfall of that, okay, sugar in the body, you take this antibody, sugar will be controlled. Or else you want to get the children at the age of 60, you take that, okay, recombinant technology, you take the sperm and recombinant technology, you make that molecule and use in future, say at the age of 60, you will get the children again. Like for example, cosmetic, you want to get the black hair, red hair, yellow hair. After 50 years, you will get the different color people, different hairs are there. Because of that responsible protein taken out, RDNA technology, use that and make the powder and take that particular powder, you will get the red hair or black hair or green hair. Or example for cosmetic item, chest up you want, no need to go for, okay, gym. You take that responsible gene, take out that antibody, and that, okay, that antibody you take out and RDNA technology, use that and make the powder antibodies. And whenever you use that antibody, your body no need to go to gym. It will automatically come up the body. Like that, in future biotechnology method, many antibodies are responsible in our body for the construction of our body. That antibodies are nothing but proteins only, keep it in mind. Antibodies are proteins only. The protein is made from DNA, DNA to mRNA, mRNA to protein synthesis. That protein is antibody. This is the mechanism. DNA, I told the God has given 23 pairs of chromosomes. That is a DNA, nothing but the DNA to mRNA, mRNA to protein synthesis. That protein is pancreas protein, for example. So diabetic, I got the age of 40. I am asking the pancreas, hello pancreas, how are you? Yes, I am fine, it is telling. But why are you not sufficiently releasing the, okay, that pancreas is not releasing the insulin molecules. Then I am asking that pancreas, why you are not releasing? Then pancreas is telling, don't ask me, you ask my DNA. Then I went to DNA. Why DNA? You are not giving the sufficient pancreas energy. Protein synthesis, you are not doing properly. I asked the DNA. DNA told to me, so many, okay, pressures are there. So a lot of stress is there in my body. I am facing a lot of stress on DNA, and DNA combination, adenine, guanine, cytosine, thymine, uracil, like this combination is twisted somewhere. It is DNA changing than regular shape. It is nothing but, okay, DNA deshaping, and it is called as a DNA mutation. So for any disease, permanent disease, the responsible is nothing but DNA. The DNA is muting. The DNA is, okay, reshaping, or it is called as a DNA mutation. That's what sugar you are getting. DNA mutation. That's what you are getting, that, okay, cancer. Different permanent disease you are getting. What is the reason means? You ask the pancreas protein not giving means, ask mRNA and DNA. There the, okay, DNA is not properly making the protein synthesis of this pancreas. That is a problem. Because that DNA is telling a lot of stress is there in the body. That's what I'm not doing. So this is an example for to make the drug in the biotechnology method. I told antibodies are more important. Our lifestyle is very important. Our life system is very important. You have to take 8 food only. But instead of that, you are irregularly taking means this also body will not accept the things only. So like that, basically drugs are two types. One is synthetic drugs. Synthetic drugs like a military, it fires that, okay, enemies. But okay, according to biotechnology, antibodies you build up in the body, then it kills the body. It kills the, body inside that can be, okay, that means microbial are there. It kills that antibodies only. That is nothing but in terms of monoclonal antibody or in terms of vaccination. What is vaccine? Vaccine means it is killed or attenuated. Killed or attenuated. So attenuated means it is nothing but killed. Killed or attenuated antigen. Antigen means any dangerous, okay, foreign body which is not required for our body. You kill it and send in the body. That is nothing but vaccine. What happens? Suppose you are taking the snake. You kill that snake and take it in the body. Then our antibodies responds very well because it feels, antibodies feels snake came and it kills our body. Then it tempts more and we are telling you are more energetic. Energetic means then antibodies are attempting to kill that antigen. That is the criteria of the, okay, this monoclonal antibodies, and nothing but body required antibodies are replaced or add more monoclonal antibodies are nothing but, okay, these vaccines is the technology. Those basically I told two types of drugs are there: synthetic drugs and antibodies. Nothing but making antibodies, monoclonal antibodies, replacement in our body. Wherever shortage is there in our body, you make the monoclonal antibody, eat it, you will get more energy. And otherwise, vaccines you take, whatever antibodies are in our body is not activating, it is becoming poor, dull. Then we are telling that antibody, you are more energetic, come on. We are telling that antibody by showing killed antigen. By showing the killed antigen to our body, inside antibodies are tempting only, which works very nicely to kill that antigens in the body. That is a phenomenon of vaccination. Very simple vaccination technique. You're indirectly promoting that antibodies to get more energy and to kill the antigens in the body. That is nothing but two types of mechanisms I told. Basic mechanism, drug discovery mechanism I told. Okay, but this is the basic mechanism I told. Now, administration of drugs, mainly eternal and parenteral. This is very simple. And topical, these are the things next. After this next slide is mine is classification of diseases. I told what is the drug. I told how to make the drug. I told basic idea. I have given the drug mechanism, either synthetic or this thing, and how many types of diseases are there for our body. That is main criteria. Like that, new diseases are coming to the body. What are the new disease means? Like corona, like different coronas, different shape. Our body is not identified such an antigen which are coming into the body. Our antibodies are not able to recognize that. That will become the big challenging job for us. We don't know the shape of the cancer molecule, cancer microbe. Cancers, okay, microbe, we don't know the shape and structure. That's what we are not able to find out the cancer molecules. That's what the alternate is, vaccines only. We can tempt the vaccines easily than synthesis of drugs only. Synthesis of drugs, it may work, it may not work, and also it is not safe and efficacy for a particular job like corona or any other disease. But vaccine is, if we know the antigen structure, that means corona structure, if you know that structure, we can make that corona and kill corona, and we can introduce into the body that will in terms of vaccine, and we can kill it. Okay, but the purpose will be survived. So therefore, different types of disease are like increasing the diseases, and we have, this is the challenge job for synthesis and biotechnology methods to identify the new drug. That is the basic drug discovery mechanism. Then first, who discovered the drug? What is the drug story means? First, homeopathy people developed the drug in 1805. Okay, Dr. Samuel Hahnemann, first he prepared the drug quinine from natural product way, from cinchona bark. He prepared the quinine drug, which is used as a homeopathic drug in 1805. Then later allopathy came in 1810. Allopathy came. He is also father for that. Allopathy also developed by him. First homeopathy, then allopathy came. After allopathy medicine, many medicines came into the line. Then afterwards, next Indian medicine, or every country depends upon our body and structure in that country. The country medicines are there. Like I have seen in Japan medicine, or in China, many medicines are there, their local medicines. And like Ayurveda, these are called Ayurvedic medicine, is nothing but Indian medicine. Our traditional medicines by any country is called as a traditional medicines are also important, like homeopathy, allopathy, traditional medicine, nothing but Ayurvedic, or any Chinese medicine, that's called as a Chinese medicine, Japanese medicine, like this is all called as traditional medicines in terms of, so now in India, Ayush department is there to take care about this Ayurvedic medicine also, that is Ayush. This is the history of homeopathy, allopathy, Indian medicine, or any country their own, depends upon their own immunity power, depends upon the own immunity power required, something for body, because hot countries are no need, because we are not affecting corona, because of hot countries, South Africans you see, nobody attacked, because of hot country. So in that, you see the cold countries, like France, that type of countries are attacked many, because of the body immunity is like that. So those home medicine is required for that. Anyway, briefly, I brief out what is drug design. Discover how to discover the molecule. I explained. Now next one is how to a drug design means. A doctor is not sufficient. Your anatomy professor is required, doctor. Biochemistry man is required. A pharmacist is required. A scientist is required. All together, they have to identify the first disease, like corona, new disease, and diagnose the disease. Then find the reasons for the disease. What are the reasons for the disease? Design the new drugs for more safe and efficacy important. The drug means sulfuric acid we cannot take to kill that corona. Even ammonia you can take in the swelling of that ammonia, which kills the corona. But we cannot take because more ammonia, because safety is not there. Efficacy is there, but safety is not there. So in chemical synthesis, safety of the drug, efficacy of the drug is more important. So therefore, in the drug design, disease and symptoms are more important. Anatomy, physiologist, biochemist, pharmacist, scientist is required. All together, they have to diagnose, they have to analyze the disease, they have to find out the thing only. And therefore, so this is drug design. Next one is, I taken the central nervous system. For example, a body, you see that central nervous system. Why I have shown the central nervous system means, I tell very briefly some anatomy and the physiology. You take that brain. The brain is in your brain. Around the brain, there is BBB, blood-brain barrier. Blood-brain barrier means you are today taking the festival more amino acids, fats, and sweets you are taking, which goes to your stomach. Every day you are taking a half kg, you are taking today 1 kg. It will go to that, okay, in the body, but it will not allow to go to the mind. Whatever the required, brain is required, amino acids, fats, and a limited amino acids, limited proteins will be allowed to the brain. Not all. So BBB controls the everything. Blood-brain barrier controls everything. This BBB, around the BBB, several neurons are there. For example, your leg fractured. That information will go to that neuron. Immediately that neuron will tell to the brain, hello brain, that my leg was fractured. Then brain feels pain. Some doctor is cutting the stomach of the woman. Then immediately that information goes to one neuron. That neuron will inform to the brain. Brain will tell it feels pain. So these all neurons around the brain are informs the new messages to that brain. Like different pains are there, either external pain or internal pains. Like for example, your teacher scolded. Then that information will go to one neuron. That neuron will give the information to the brain. Then brain feels, okay, my teacher scolded like that. Or in our family member, our mother expired. So that information will go to one neuron. That neuron will give continuously information to that brain. Brain always feels what happens, my family member expired, like that. It feels very bad. Then it's after that cell, after that protein, in the brain, some part of the protein can be destroyed because of the continuous thinking, continuous disturbance of that part of the tissue and the protein, it disturbed, it converts to tumor. The regular cells are healthy cells, healthy cells because of that force, it converted to tumor cells. Tumor cells converted to, okay, cancer cells. This is the regular phenomena. These regular cells are, healthy cells are, sometimes because of the strain, because of that, okay, unwanted, uncontrolled things, uncontrolled growth happens from regular cells to uncontrolled growth. That growth causes for the tumor. The tumor is next stage converts to the cancer. This is nothing but brain cancer. For example, I am telling, because all problems are coming different, okay. Neurons are continuously giving the pain means brain gets a lot of pain. Therefore, it requires a lot of strength to recover all the parts of the body mechanisms to control that. But it takes some time also to command that brain to the parts of the body. If something happens, it takes little time to control that also because it's giving the order slowly. So I am telling central nervous system you take. This is the basic skeleton in the body. This central nervous system is a bridge or connection to, just like our roads are connected to Delhi to South India, way to any place. Like also any information, any pains in the body happens through central nervous system. It reaches to neuron. Neuron is BBB. Around the BBB, particular neurons are there. That information goes to the brain. That then only mainly mechanism happens. Then it feels, I have cancer, I have diabetic, I have pain, I have external pain, somebody beaten, my leg was fractured, my finger was fractured, my knee was paining. Like, okay, all different kinds of pains or diseases are recognized by these neurons, and which gives the information to brain, and brain controls and orders that, and you take the medicine. When we go to medicine, suppose when doctor want to operate a lady, human being, sorry, a woman, then doctor will give the medicine, because without medicine, this information will go to neuron. Neuron immediately tells that brain. Then our doctor cannot do that operation. We will feel pain. Then tablets we will take. That tablet will go and will bind with the neuron, which neuron is giving the information, doctor information to that brain. That neuron will be, okay, he was blocked by, that neuron will be blocked by this tablet only. This tablet works 8 hours. Till 8 hours, doctor can kill, doctor can cut the operation, doctor can do anything. He can put scissors inside, he can, okay, everything he can do. Because the tablet works to that time. Like all pains are required, all kinds of tablets only. We don't have sufficient, that our family member expert continuously we are getting pain, that converts to that, okay, cancer. I'm telling, if immediately medicine is there, which can be cured, but we don't have such a neuropeptides. These medicines are called as neuropeptides. This medicine is peptide. Neuropeptide medicines are, a lot of peptides are required to control the pains of the body. Once you control that peptides, by taking the peptides, you will not get the cancers, you will not get the diabetic also. Such a different external or internal pains can be controlled by this neuropeptides. Still neuropeptides are rising, that synthesis is going very well. This is, I told, this is in the drug discovery program. What is the drug role I told in the body? Okay, this is, you can understand, I think. Are you able to get my voice? Are you getting my voice, doctor?
Proceed, sir. Proceed.
Yes.
It is
Yes.
very
Next
clear.
one is, so after that, I told already, DNA to mRNA, mRNA to protein synthesis via transcription, translation. This is continuous process in our body. You are making disturbance for conversion of transcription and translation, means it started getting diseases for our body. When we are getting the problems in our body, you are disturbing transcription and translation. You are not taking the nutritional food properly, required to clean or to enhance the body immunity for all different liver, heart, or different parts of the body required a nutritional food and nutritional mechanism, and actually real mechanism which will help for that. You will, your body is healthy body. Otherwise, your body is not healthy body. So therefore, all these controlled by translation, transcription, structural framework of the cell. Structural framework of the cell means cell is healthy, your body is healthy. Your framework always you have to take care by yourself. You should not disturb your cell. Some proteins act as enzymes, catalysts, and catalyze the biochemical pathways. So therefore, cell is more important, mono unit in our body. Cells are acting as a, okay, body mono units like a bricks in the building, or else it is enzymes to catalyze the biochemical pathways in our body. These some proteins helps in the transport across membranes. Proteins act as oxygen carrier. Some are storage proteins. All membranes are lipoproteins. You see the protein importance in our body is more. The protein is made up with cell. Cell is made up with from DNA. DNA to mRNA, mRNA to protein. Protein synthesis is nothing but cell only. The cells are structural framework. Some proteins are enzymes. Some proteins are transport across membranes. Some proteins are oxygen carriers. Some proteins are storage proteins. Some proteins are lipoproteins. All membranes are lipoproteins. Our membranes, body membranes are lipoproteins, which are safe to the body. Like protein is very important. So I mean to tell, our body cells are more important. If you take care of the health of that one, you are okay, you will not get any diseases. Okay, this is all I have given. All I have given. Next slide. All I have given the okay, importance of drug discovery. The basics of drug discovery I explained till now. Now I want to I want to tell something about the cancer. Now till now, what is the drug discovery? How many types of drugs are there? How to make the drug? What is the drug mechanism in the body? Basically, how it acts in the body mechanism. How that immune system will be enhanced in our body. By taking the vaccine, immune system can be raised. Simply vaccination is not a medicine which can enhance the antibodies power or immune system. You have to remember the entire mechanisms here. Two types of drugs I told. Now come to the point of cancer. Why you are getting the cancer? Why are you getting the coronas and some other new diseases? I want to explain that. Now, cancer is very simply an abnormal cells division in the body. Normal cells are abnormally started dividing. Normal regular cells are abnormally dividing uncontrolled way. Abnormally dividing and uncontrolled way it is growing. Normal cells. All of a sudden, something happened to that cell. As I told, you are not protecting that healthy cells. Healthy cells are, every day you are not giving proper food. What happens? It will become unhealthy. That unhealthy cells after some time, it will become the tumor I told. In that position, when it is converting into tumor cells, it abnormal cells starting and uncontrolled growth will start. After making it, then this tumor is nothing but a fracture in the body or something disturbed our body. You cannot, like a rice is disturbed after 2 days, you cannot get back your rice. Like also our part of that body is disturbed like to tumor and tumor to final cells. You cannot get back your cells. When it is in the tumor stage, you can cut that cells and you can remove the tumor cells. Then only you can, you can cure that cells. Then only you can control the cells. There is no exact medicine for this. So this is the first time I am defining the cancer cells. What is the cancer cells? How it starts the growth means it is a disturbed. You are disturbing your cells. Then you are getting the liver cells you are disturbing. Okay, suppose, for example, different types of cancers I will tell. First of all, you are disturbing the cells and it is, you are inviting that cell, disturbing and inviting the tumor cells. The tumor stage, okay, you can remove, and after tumor stage, you cannot remove, and these cells are, you have to cut and remove, or you have to laser treatment you have to do to kill that cells, then only which can be cured. Anywhere, that cancer is, different types of cancer is carcinoma cancer. This is the skin cancer. Or in the body, entire body is connected with the tissues. Body is, I told, based on cells only. Cellular mechanism in the body. And tissues are connected to the cells. One cell to another cell is connected tissues mechanism only. Any body parts are made cells and these are connected. One part to another part is connected by tissues only. Then skin, basically skin I told, membrane, protein membrane skin. That protein membrane is, I told the protein, that protein was disturbed. Then that skin was disturbed. Then automatically that is called as a carcinoma. Carcinoma. Okay. Then sarcoma. Cancer that begins with the connective or supportive tissues. Only skin and tissue is carcinoma. Connective tissues. I told connective tissues, body part to part 2. Supportive tissues, such are disturbed, are bone, fat, muscle, blood vessels. These are disturbed means that is called sarcoma cancer. Sarcoma cancer was caused by connective tissues, supportive tissues, bone, cartilage, fat, muscle, blood vessels. These are the different types of parts are disturbed in sarcoma cancer. And next one is leukemia cancer. In the body, the blood tissues, the blood is disturbed. The blood cells are disturbed. That means blood proteins are disturbed. Then the cancer starts in blood forming tissue. When blood forming tissues, that cells are disturbed means that is the leukemia cancer. Because of the bone marrow system. The bone marrow system is a basic thing to develop that cells. That disturbed means you will get the blood cancer. Blood cancer is because of that only. Next one is lymphoma. Lymphoma or myeloma. Lymphoma and myeloma cancers. Cancer begins with the immune system in the body. The body construction is immune system is more important, I told. Antibodies are perfect. In the body, all antibodies are healthy. Your immunity is more health. You will not get any diseases. You will not attack by any diseases. So, immune system is perfect means then it is good. That is, if it is not good, lymphoma or myeloma cancers will come because of the immune system damaged. You are not taking the proper food for immune system means you will get the lymphoma and myeloma cancers only. And next one is the brain cancer. As I told, you are taking a lot of problems to the brain. Every day you are so many tensions in the brain, and that brain causes for, okay, that cancer is called as a brain cancer, which is connected to brain is connected to always spinal cord cancer. Spinal cord and brain are connected. Central nervous system is connected. That is all under brain, spinal cord concerns or cancers only. These are now central nervous system. So in the body mainly skin and tissues are there. That's called carcinoma. Attacked by carcinoma, cancer, sarcoma cancer is connective tissues, supportive tissues are in the body. Bone and cartilage, fat, muscle, blood vessels, sarcoma cancer, and blood forming bone marrow is disturbed means leukemia cancer will come. And next one, immune system in the body is disturbed means you will get the lymphoma or myeloma cancer. Next one, central nervous system is disturbed. As I previously shown the slide, central nervous system is disturbed means brain cancer or spinal cord cancer will come. So therefore, basically our body is okay, connected with the different types of these tissues, these cells only, central nervous system only, bone marrow only, and different cell system only. Because of that parts only, you will get the different types of 5 types of cancers only. So that's what these are the types of cancers. Okay, so this you can understand that cancers, what type of cancers. Now, so this is the mechanisms for to treat the cancers. What type of drugs are came into the market till now? Means I told like different nibs are there. Erlotinib, sorafenib, imatinib, oxatinib, cisplatin, capecitabine, and gefitinib, gemcitabine, lenalidomide, sorafenib, melphalan, bendamustine, erlotinib. Different types of molecules are developed by the people. How they developed means from known to unknown. That cancer cells in the body, that patient may live 6 months or 1 year. He may die definitely after 1 year. At the stage of 6 months, as I told, in tumor state, you can use our molecule. That molecule kills that tumor cells only, not cancer cells or little cancer cells. 10% or 20% cancer cells can be cured. Or else you have to cut that part. For example, for ladies, that okay, brain cancer. For example, this chest cancer is there. The chest cancer means you have to cut the chest cells that you can save. Or else any part of the body is disturbed means you have to cut only. But all parts are not possible. Blood cancer not possible. Digestive system not possible. You cannot cut it. Like so, you need to develop the drugs only. Like in this mechanism, platinum compounds are there. It worked as a platinum is powerful metal. It acted as a cancer molecule. In the structure, more nitrogens are there, more fluorines are there. Whatever I explained, all the molecules, in that molecules, fluorines are there, chlorines are there, more nitrogens are there. In that structures, those are the inserting, those nitrogens, and those things you are inserting, and it is becoming a molecule for cancer, which kills that initial stage cancer cells only, not all stages. And if it is in the second stage, the cancer cells can be killed by radioactive methods, or sorry, by therapy methods. That is nothing but your laser treatment. You are giving a laser treatment that cancer cells can be killed automatically. The cellular system, wherever the cancer is disturbed, that can be killed, that can be removed permanently. Then only the patient survives only. There are the 2 types are there. Initial stage, you find out tumor stage, then take the medicine. It is okay, or it is 10%, 20%. Then you take that, kill that cells up to 50%. Then possibility is there. At the time, you have to develop the immune system. You have to more confident. You have to rich in the mind, but more resistance power you should make in the body. Then you can survive. If you feel every day, I got cancer, I am going to die means soon we can die. We should get more confidence and more immune system. You should develop by taking the more natural products, more immune developed centers, more nutritional food, which can require for our body to clean and require exact food for our body that you have to take, not medicines. Natural foods are more important to cure the cancer. Next one, we have developed some molecules and we screened docking studies we have done and we applied for cancer, and some are active and we published in the very good journals like ChemCom, Medicomcom, or European Journal of Medicinal Chemistry, and in the top journals we have published these molecules. Still, I am continuing that cancer molecule synthesis, and we are screening for anti-cancer. Different anti-cancer depends upon the cell system, and we have developed so many molecules in for cancer, and still we are developing the cancer. Some American cancer institutes, they are supporting us, and they are writing letters also to us to send some cancer molecules to them screen. Recently, for pharmacy students, I tell one Malaysian institute, that is International Medical Research Center, IMU, International Medical University, pharmacy-based university. Doctors, pharmacists are there. They also, we have tie up with that, our company. I am the director or CEO for this, Dr. Max Pharma. We are synthesizing some molecules, we are sending for them. There, one pharmacy professor, head of the dean, which we are sending to him, he is screening. Very nicely, that students also planning to come here, and we are sending some molecules for cancer screening. Next, we have synthesis. This unfortunately, these slides you are not able to see. I'm very sorry for that. And these are the screens, different synthetic methods I'm showing in the screen. All I'm changing, just I'm putting the trifluoromethyl groups, monofluoromethyl groups, or more nitrogens, or more oxygens. Those things I am putting in the structure. That only I am doing, not mimic. This is very simple technique. Known to unknown. Already known drug is there. Insert some non-metallic species and chiral center, aromatic center, 5-membered ring, chiral molecules with fluorine groups like CF3CO group, single fluorine, hexafluorines. Such a molecules like heterocyclic rings or alicyclic rings. Okay, so that is rings 5. These are I'm inserting in the molecules. I'm making them making. That is the only research we are doing, sir. Next.
Sir, continue, sir. Continue.
Yeah. So now, okay, now the DNA mutation. Why you are getting the cancers? Means your DNA, healthy DNA is muting. I told no. I asked that pancreas, hello pancreas, you are not able to make the sugar molecules, insulin molecules to kill our sugar, excess sugar. I'm taking one day half kg sugar, one day 100 grams. 100 grams sufficient to body, but 1 kg in the function I am taking, surplus in the blood. That sugar cannot be cleaned. I asked pancreas, means it was not able to do cleaning because the protein is not 100 molecules of insulin not making, 50 only making, then that is type 1, 100 not making, type 2 diabetic. The pancreas is weak. That is asked by DNA. DNA is telling I was muted and DNA mutation happening. Like basically DNA mutation is a cause for cancers. Second one is hereditary. Some cases hereditary may come, not all the cases. Your father have cancer, but we take healthy body, we control our body. Depends upon all healthy body depends in our hand only. Okay. So next one is acquired. Due to natural systems, you are acquiring from surroundings. That is called acquired. There are the 3 types of ways: DNA mutation, hereditary, acquired. That acquired means radiation may cause, viruses may cause, chemicals or toxic metals handling may cause, drugs may cause. These only reasons for cancer. DNA mutation for pressures, because of the pressures, lot of pressures. Hereditary is cause. Radiation is cause, acquired. Viruses, radiation means alpha, beta, gamma, radioactive metals are there. You put alpha, beta, gamma, those are inserted to the body, affected our body by that radiation, you will get the cancer. Viruses, different viruses, by chance it is going inside, you may get the cancers. And chemicals is the toxic chemicals which may cause, and drugs are there which may cause, some drugs are there dangerous to the body and which may cause for cancer. Next one, next. Like this, the purines, pyrimidines, we are making some peptides. Like all, we are making the molecules and one peptide, like different amino acids I'm coupling to make the DNA supporting molecules. DNA repairing is there now. That repair molecules to support the DNA repair, some molecule are synthesizing like amino acid tie up with the fluorine compounds or chlorine compounds or nitrogen containing synthesized compounds are coupled with the okay, these are peptides like amino acids, and these are more close to our DNA. That's what it is going to our DNA, and disturbed DNA may be repaired. That is the aim of our cancer molecule synthesis. DNA repair will give the good direction for okay health only. That's what these molecules are. Okay, we are making for that. Next. Therefore, you can see in the slide radiation, different pollution, like chemicals carcinogenic, including smoking, mobile phones, injuries, like hereditary factors, immune system affecting, virus infections, and stress only. The biggest thing is stress only. That's what I suggest the medicine. The best medicine is like, okay, you use that, okay, that is every day. You do that, your brain should feel okay, relief, should feel happy. While sleeping also, it should be rest is need for these cells only. That is nothing but around the brain, the cells should be taking rest. It be healthy cells, then only it will become health. Then only for that you take the meditation or yoga. Meditation will give the good health for your BBB will clean every day. Blood brain barrier will be cleaned every day by doing yoga or meditation. Then it will become active. Then it functions very well, which gives a good effect to that good work to the brain, which can control our diseases. Mainly, yoga and meditation is the good thing for the cancer patients and for any other patients and any for other stress also. These things are needed than tablets and capsules. So I told this drug discovery program, you have to synthesize the molecule and non-clinical development. First, you haphazardly you have to check in the lab, then animal studies. Without animal studies, that is called non-clinical development. With animal studies, second next phase, the phase 1, phase 2, phase 3 is there. In the animals, phase 1, like rats, phase 2, also animals, phase 3, human beings. In that also checking. These are the okay drug discovery program. First, pharmaceutical synthesis chemistry, then analysis, purification, then third one is okay. So that third one is nothing but Sutix, making tablets. Sutix. Third one is nothing but this is pharmacology, screening is okay. Then you have to FR&D and synthesis of the tablets and capsules, FR&D studies, stability studies, those part will be there. This is the entire system is required in R&D and drug development program in the company. These things are required. In vitro studies, cell lines, cell-free systems, drug screens, drug formulation, chemistry, manufacturing and controls, in vivo efficacy studies. In vivo is nothing but animal models and proof is in vivo. In vitro means cell lines and cell-free systems. Drug screening only, in vitro. In vivo means naturally on animals. Animal models and proof of principle. Non-clinical safety studies. Finally, safety studies are required. More efficacy and safety is required for the new drug development. This is what we are making so many molecules, and among those many molecules, only these few molecules will work out. Next. So this is GMP systems, drug supply issues, drug formulation issues, and preclinical pharmacology, and clinical pharmacology, early phase. These are the challenging jobs in this thing. This is about the brief lines about the drug discovery program. So this is content, general content in the drug discovery program today. This I will stop here. If you have any doubts. And second part is the catalysis part. Also, we work on catalysis. We prepared many catalysts in Japan, and I worked in Japan, many catalysts, new catalyst I developed. And then in Korea also, we developed the new catalyst. And in Taiwan also, I developed the new catalyst. In France, Strasbourg University. Presently, I am working with the Australia, Queensland University tie-up. We are developing some molecules, materials, new materials for cancer. Also, recently we got some fund for cancer our DBT, Department of Biotechnology versus Australian government. We take some mesoporous material, antibody we keep in that mesoporous material, and on the cancer patient, the blood is there. 10 grams blood I will take cancer patient. I pour in the antibody, which is kept in the mesoporous material, and 10 grams 10 grams, 10 molecules are there, microbes, cancer cells. 10 are attacked on antibody, 100% patient is suffering. Out of 10 grams, 1 molecule attacking on antibody, 1%, 10% is suffering. 2 molecules are attacking, 20% is suffering. 3 molecules attacked out of 10, 30% is suffering. Our project is, we have written the project without knowing the cancer in our body. Many people are dying until 80%. We are not known. That's what we are dying, many people. We have to make an instrument to check the cancer. That's what we need fund means. Australian government and myself, we wrote for our company, we got some fund. We are developing some antibodies which can keep in mesoporous material, and the cancer blood we are pouring in that method, that cancer cells are attracted by antibodies. How much attraction is there, that can be calculated. How much percentage of they are suffering, we can calculate by that biomedical applications we are doing now. So this is the cancer, okay, screening in what stage that we are doing. We got some fund also from Australia and DBT that we are doing at present in our laboratory, in our company also. Next. So this is about the cancer program. Apart from that, in our company, we are making anti-cancer intermediates, anti-cancer molecules, new molecules. From America, they give the new cancer molecule, NCE, new chemical entities, 10 steps, 12 steps. One big project we have done for America. They given the 12-step molecules, 100 kg to 1 kg we prepared. We submitted to them. They spent nearly 12 crores. And I don't know, they have spent 12 crores means that is cancer molecule. They are screening now. Phase 1, phase 2, phase 3 screening they are doing. Outsourcing they have given to us. Such a cancer molecules we are doing. At present, we are getting some molecules from Europe also. In future, we are going to do many cancer novel molecules, new chemical entities, and giving to others only. So this is the program. Next, these are some of the pictures with Nobel laureates. I worked with some Nobel laureates. That pictures are there here, but unfortunately, you are not able to see. And thank you for all the listeners and given opportunity to JNTU means I'm very happy to take the lectures at JNTU. Basically, 8 years I served in JNTU, 95 to 2003 in JNTU, Hyderabad. I worked as a lecturer along with my best friend, Dr. Devanna, Professor Devanna. He is my best friend, and we together worked in Hyderabad. And so whenever you call me, I am ready to give lectures for Anantapur JNTU. And also I studied my MSc Anantapur JNTU. JNTU I studied. I studied my MSc SK University Anantapur. I have that connection. Anytime that I love that Anantapur JNTU, and this principal is very good. I want to learn that, that meditation and all things with him when I get the chance. But one time only I went to him and he taught some lessons for meditation that is for everyone required. I like that program. He implemented entire college that is needed for everyone. Apart from all our studies, that needed for everyone. So I request again that principal to implement in our college. That is the best medicine you are giving to the people, to the students. In that age, you have to give to them. You are doing that is the best thing. In that age, they learn something like this is their body will be the healthy body. I feel that. Please do it. And we support always to you. And to entire the JNT system, I am very thankful to them for giving this opportunity. And thank you to principal.
Thank you, sir. Thank you.
Thank you.
Sir, Dr. Chari, sir, an excellent session. But the only thing is that we missed that one in the slides.
Yes, sir.
But even though you covered all the points and very basic aspect to the up to the level, how the disorders and diseases getting.
Yes.
And any questions, please, already you are there in next session will be there. Participants, if any questions will be there, 1 or 2 questions, or otherwise, and we'll give the vote of thanks to the today's speaker, Dr. Chari. Then we'll start the next session.
Yeah.
And Pranati.
Yes, sir.
Okay, ma. Please give the vote of thanks, Pranati,
Okay,
Dr.
sir.
Chari.
Thank you, Adarvana Chari, sir. You gave such a very valuable and inspirable speech to us. You covered most of the subject platforms such as biotechnology, medicinal chemistry, organic chemistry, microbiology, anatomy, and pharmaceutics in the time of given to you. It is not a normal job. You explained drug discovery process step by step and how to overcome problems which are generated during drug discovery process. by taking proton pump inhibitors, anti-diabetics, anti-cancer drugs, and vaccines. We got a clear structure of immune system of human body and how cell parts were involved in immune system to treat a abnormal environment in our body. After hearing your session, we understood why cancer comes, what will happen in human body after getting carcinoma, what are drugs available in the market to treat cancer, what we need to design novel drugs which acts as anti-tumor, and how to develop them. Adarvanachari sir, you said all those valuable topics by taking examples that are natural and understandable. Definitely, we will follow your guidelines in our research career and teaching career. From the bottom of the heart, we are very thankful to you, sir, for giving such a great session and for sharing your experience of research with us. Thank you, sir.
Thank you very much. Thank you very much.
Thank you, Pranati. And thank you very much, sir, Dr. Adarvanachari, sir.
Thank you,
again,
sir.
we'll invite you for the session for the PG students.
Right.
Okay, thank you very much. Thank you.
Thank you, sir. Thank you very much. Thank you.
Okay, next session, Sailaja, Dr. Raghavendra.
Sir, good afternoon, sir.
Good afternoon. 1 minute, Sailaja. Wait, please wait. Okay. And now Dr. Raghavendra, a warm welcome to Dr. Raghavendra, the manager and quality control department, head and manager quality control department, Dell Excel Pharma, Hyderabad. And we are inviting him and the FDP program sponsored by AICT Jntua Anantapur to deliver a talk on this one, what the quality aspects. And Ms. Sailaja.
Sir, yes, sir.
Yes, please introduce the speaker to the all the our faculty fraternity.
Okay. Good afternoon to everyone. Myself, Shailaja, faculty of pharmacy in Jntua